Hello everyone, welcome to our channel Pharmacotherapy Made Easy. Today we are going to discuss a topic on drug discovery. So what is drug discovery? Drug discovery is a process in which we will be identifying a drug molecule that will be used later in the later phases for treating different diseases. So first of all, before discovering a drug molecule, a disease will be selected. In that disease, the major mechanism that will be responsible for the causation of the disease will be identified. After identifying the cause for the disease, we will identify a target. This process is called as target discovery. Once we discover a target, we need to identify some molecules which can act on those targets. So this process is called as target identification or validation. There will be different tools to identify these targets. Usually there are tools like functional genomics, structural genomics, proteomics and bioinformatics. These types of tools will be used to identify these targets. Once they identify the target, they will validate the target. Is this the real target or did we discover any fake target? They should know whether this target is the real cause that is causing the particular disease. Once they identify the target, they will check different molecules. The big pharmaceutical companies will be having a libraries of molecules. From those molecules, they will check whether the molecule is going to the target and binding to the target, whether the molecule can change the target's rotation or any other thing. They will identify this. This identification process is called as lead identification. So lead identification will be done using different techniques like high throughput screening, in silico structure based drug design, like that. They will use different tools. This process is called as lead discovery. Once they discover a lead, then they will optimize the lead. What is lead optimization? In lead optimization, they will develop this lead into much more drug-like thing so that it can be used for the treatment of patients. So first what we are doing in drug discovery, it's like a big umbrella. Under that there are many specialities and other things. The first thing comes in disease mechanism. First we should know clearly the disease mechanism. Once the disease mechanism is elucidated, then we will identify the target. This process is called target identification. Once we identify a target, the target will be validated. Once the target is validated, then comes the next step, testing different molecules that we have. There will be some hundreds of thousands of molecules within the molecular libraries. From these molecules, they will check the action of the molecules that the company is having on the target. Their activity will be identified among the large number of molecules. Some molecules will be identified as leads. This process is called lead identification. Once the lead is obtained, some leads will be around 5,000, like that. Let us think 5,000 leads we have obtained. From those leads, only some molecules are much suitable for the drug to make it as a drug. This is called as lead optimization. Finally, we'll be getting hits. These hit molecules are the major molecules that will be used for drug discovery. After this process, when they identify a hit molecule, finally they will decide certain molecules that should be used. These molecules, first they will use these molecules in animal models. The testing of drugs in animal models is called as preclinical studies. So during preclinical studies, different animals like rodents, mice will be used. Some even use rabbits, dogs. Based on the drug and the disease, they will choose which animal model to be used. This stage is called preclinical study. After this preclinical study, the drug enter the next stage, that is clinical trials. The clinical trial, it is called as clinical because it is being done in humans. Any testing that is being done in humans is called clinical. So clinical trial contain three different phases: phase 1, phase 2 and phase 3. So what do they do in phase 1, phase 2 and phase 3? Briefly, simply to tell, in phase 1 they will study PK/PD, they will assess the tolerance, they will assess the pharmacology of the drug in phase 1. Phase 1 would be done in healthy volunteers, whereas phase 2 and phase 3 will be done in the patients. Phase 1 will be done in healthy volunteers, whereas phase 2 and phase 3 will be done in patients. Phase 2 can also be called as therapeutic exploratory studies. In this, they will identify whether the drug will be useful for the patients or not. Next, During phase 3, it is called therapeutic confirmatory studies. What do they study? They will confirm the drug efficacy. They will identify whether it is safe. Since a phase 3 study will be done in around nearly more number of patients when compared to the phase 1 and phase 2, they will take around 5,000, 3,000 like that patients in phase 3 trials. So the sample size will be high. The number of adverse effects identifying will also be high, so that they can assess benefit risk relationship also during phase 3 studies. After these clinical trials, after phase 3, if it is confirmed that it is safe and effective, the product will be launched in the market. Once the product is launched, the final phase, it is being called as phase 4 trial nowadays. This is post-marketing surveillance. What is post-marketing surveillance? During post-marketing surveillance, once the product is launched in the market, people and drug doctors or whoever healthcare providers or patients will give us a feedback on side effects. This is called as post-marketing surveillance. The major stream that is coming in nowadays is pharmacovigilance. Pharmacovigilance is related to post-marketing surveillance, and some side effects are also identified during clinical trials also. It also comes under vigilance only. And when we come to job opportunities in drug discovery process, there will be many job opportunities, but for pharmacy and clinical pharmacy related students, the job opportunities which are more feasible and available are clinical research coordinator and clinical research assistants or associates. These jobs will be available in clinical trial phases. Here we can be working, and we can even work in hospital setting where the research is being done on drugs on human beings, that is clinical trials in hospitals. And we can even work in a pharmacovigilance sector. Whereas other domains, they are also useful, but it's not much relevant to clinical aspects. It is much relevant to organic chemistry, MSc organic chemistry, and other approaches can be done, whereas M-Pharmaceutics can also be entering these studies. And finally, when it comes to registration, once all these studies happen, after phase 3, we will be applying for new drug application, whereas during in between, we'll be applying for investigational new drug before it comes to human itself. That is called registration process. In that registration, regulatory approach, depending on the country and area where the drug is going to be marketed, the regulatory approach will be differing. And post-marketing surveillance I have already explained. This is an outline of drug discovery. Slowly we will be entering into each phase and each thing completely in our next videos. Thank you.